The overall goal of this affinity capture protocol is to isolate endogenous protein complexes from mammalian cells.
This method can help answer key questions in cell and molecular biology, such as which proteins form together into physically linked functional modules. The main advantage of this technique is that numerous physical interactions associated with the target protein of interest may be observed simultaneously.
Grow 1 to 8 grams of cells as described in the text protocol. Pour off the growth medium into a large beaker. Then, place the culture dish on ice in a large rectangular ice pan. Add 20 milliliters of ice-cold 1,X-phosphate buffered saline, or PBS, to the culture dish, and release the cells from the dish using a large cell scraper. Transfer the cells to a 50 milliliter tube, pre-chilled on ice. Add an additional 10 milliliters of ice-cold 1X PBS to the same dish. Collect the remaining cells and transfer them to the 50 milliliter tube. Repeat this process for each dish. Cell suspensions from different dishes may be combined to reduce the sample number and plastic waste. Next, centrifuge the samples for 5 minutes at 1000 times G and 4 degrees Celsius. Following centrifugation, carefully pour off the supernatant. Resuspend each pellet in 10 milliliters of ice-cold 1X PBS. Consolidate the resuspended pellets at up to 5 per 50 milliliter tube. Wash the empty tubes with 10 milliliters of PBS, add the solution to the consolidated samples, and centrifuge as before. After removing the supernatant, resuspend the pellet in 10 milliliters of ice-cold 1X.